Good evening, everybody. Myself, S. Ramachandran, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Vistas, Chennai, is going to give a seminar on the topic Carbohydrates and its Metabolism. Before going on to the chapter, we have to see what is the daily intake of food which should be done for any individual. The energy is nothing but 8,700 kilojoules, protein 50 grams, fat is 70 grams, carbohydrate is 310 grams, sugars is 90 grams, sodium is 2.3 grams, dietary fibers 30 grams, and saturated fatty acids. So this is a balanced diet which you're supposed to take every day, out of which your carbohydrate is 310 grams. So what are carbohydrates? They are broadly defined as polyhydroxyaldehydes or ketones and their derivatives, or a substance that yields one of these compounds. It is composed of carbon, hydrogen, and oxygen. The functional group present include the hydroxyl group, and O's indicates the sugar. Carbohydrates are the most abundant of all the organic compounds in nature. In plants, energy from the sun is used to convert carbon dioxide and water into the carbohydrate glucose. Many of the glucose molecules are made into long chain polymers of starch that store energy. About 65% of the food in our diet consists of carbohydrates. Each day, we utilize carbohydrates in food such as bread, pasta, potatoes, and rice. Other carbohydrates called disaccharides include the sucrose, table sugar, and lactose in milk. During digestion and cellular metabolism, carbohydrates are converted into glucose, which is oxidized further in our cells to provide our bodies with energy, and it provides the cells with carbon atoms for building molecules of protein, lipids, and nucleic acids. In plants, a polymer of glucose called cellulose builds the structural framework. Cellulose has other important uses too. The wood in our furniture, the pages in our notebook, and the cotton in our clothing are also made of cellulose. What is the function of carbohydrate in cells? They are the major source of energy for the cell, major structural components of plant cell, immediate energy in the form of glucose, and they reserve or store energy in the form of glycogen. So you can see the different structures of your carbohydrate present over there. Coming on to the classification of carbohydrates, they are classified into three major categories. Number one, the monosaccharides, the oligosaccharides, and the polysaccharides. The oligosaccharides are three types: disaccharide, trisaccharide, and tetrasaccharides. Monosaccharides are simple sugars or the compound that possesses a free aldehyde CHO or a ketone group or two or more hydroxyl groups. They are the simplest sugars and cannot be hydrolyzed further into smaller units. Monosaccharides contain a single carbon chain and are classified on the basis of the number of carbon atoms they possess, such and as aldoses, ketoses, depending upon their functional groups. Here you see the classification of carbon atoms: triose, tetroses, pentoses, hexoses, where you can find out the aldose sugar and the ketose sugar names also. This is a classic example of glucose, galactose, and fructose with their ring chain. This is the Keller-Killiani synthesis, where you increase the carbon chamber one, therefore you get a different carbohydrate structures in the last section, like the allose, ribose, the glucose, fructose, mannose, galactose, etc., etc. So you have a very important term in carbohydrate, which is called as a reducing sugar and the non-reducing sugar. Carbohydrate with free aldehyde group as a carbon 1 or free lactose group in the ketone group carbon 2 is said to have reducing sugar properties. Aldehyde and ketone group is not free, therefore it is a non-reducing sugar. In reducing sugar, they are in hemiacetal hemiketal form, whereas in non-reducing sugar, they exist as acetal or ketal form only. This reducing sugar, they exhibit mutar rotation, whereas non-reducing sugar does not exhibit mutar rotation. The example of reducing sugar are glucose, fructose, lactose, maltose, and cellulobiose, whereas the non-reduced sugars are sucrose and glycogen, inulin, etc. Coming on to the next section of the classification, which is called the oligosaccharides. It contains the disaccharide, tetrasaccharide, tetrasaccharide, pentasaccharide, etc. The disaccharide include the sucrose, lactose, maltose, galactose, etc. The trisaccharide includes the rhamnose, the dentionose, the raffinose, etc. The tetrasaccharide includes the stachylinose and the scodulose, and the pentasaccharide includes the verbasone. Coming on to the next one, which is called as the polysaccharide. They are containing 10 or more monosaccharide attached together. Examples, a classic example is the starch and the glycogen. Long chain of liquid forms this polysaccharide. Cellulose gives structure of both to plants, fiber to a diet. Glycogen is an energy storage sugar produced by animals. Liver cells synthesize glycogen after a meal to maintain the blood glucose level. So this is an example of polysaccharide. It present in starch. We have the amylose, the amylopectin structures. Therefore, you have the two types, homopolysaccharide and the heteropolysaccharides. Structure of the starch, which has contained the amylose and the amylopectin. Amylose is a straight chain, whereas amylopectin is a branch chain. You can see the structure present over there. Cellulose is a beta chain of your glucose molecule, whereas your starch is an alpha chain. So glycogen, they are storage forms of glucose in the body, stored in the liver and the muscles. So the concept of carbohydrates is fully depicted over here as a tabular column, which clearly states what is a reduced sugar, what is a non-reduced sugar, etc. Polysaccharide, amylopectin, amylose, cellulose, etc., etc. The biochemical or biomedical importance of glucose are, as I told you before, they are a major source of carbohydrate. It's a major fuel for tissues. It converted into carbohydrates, ribose in nucleic acid, galactose in milk. They form glycoproteins and proteoproteins, etc. Coming on to the metabolism, the most important one. So the type of metabolic pathways is catabolic pathway, anabolic pathway, and the amphibolic properties or anti-amphibolic pathway. The stages and the phases of metabolism are primary, secondary, and tertiary. So you can find out food molecules, simple molecules forms amphibolic, goes down to two types, anabolic and the catabolic. Anabolic, it forms the protein, carbohydrates, lipid, nucleic, etc. And the catabolic is formation of carbohydrate and water. The major pathways of carbohydrate metabolisms are given over here in the straight chain part. One is glycolysis, citric acid cycle, gluconeogenesis, glycogenesis, glycogenogenesis, hexose monophosphation, uronic acid pathway, galactose metabolism, fructose metabolism, and the amino sugar metabolism. So what happens to the glucose and enter the cell? Insulin dependent transport system of glucose, not dependent on hormone insulin. This is operative in the hepatocyte, erythrocyte, etc. Insulin dependent transport system occurs in the muscle and the adipose tissue. So coming on to the glycolysis, which is otherwise called as Embden-Meyerhof pathway. It is defined as a sequence of reaction converting glucose or glycogen into pyruvate or lactate with the production of ATP, either anaerobic condition or aerobic condition. This is what is the salient features of the cell. So the reactions of glycolysis is energy investment phase, the splitting phase, and the energy generation phase. So this is a cycle which undergoes in the glycolysis. Glucose form glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-diphosphate, dihydroxyacetone phosphate, glyceraldehyde 3-phosphate, 1,3-bis-phosphoglycerate, 3-phosphoglycerate, 3-phosphoglycerate. Then it forms the phosphoenol pyruvate and the pyruvic acid. Therefore, in aerobic respiration, it forms the pyruvic acid, and in anaerobic respiration, it forms the lactic acid. So this is the classic example of the ATP production, which takes place in the glycolysis. Coming on to the next one, which is called the citric acid cycle, or the Krebs cycle, or is called a tricarboxylic acid, essentially involves the oxidation of acetyl-CoA to carbon dioxide in water. This cycle utilizes two-thirds of total oxygen consumed by the body. So it is a cycle chain. This is a cycle of your citric acid cycle, where acetyl-CoA is required and it being forming a cycle form. The next one is called the importance of gluconeogenesis. The term is there, gluconeogenesis. Therefore, synthesis of glucose from non-carbohydrates, which is called as gluconeogenesis. The major substrates are your lactate, pyruvate, glycogenic amino acid, propionate, etc. as your precursors for your gluconeogenesis. So it is a complete reversal of your glycolysis cycle. Complete reversal. Next comes the Cori cycle, nothing but synthesis of glucose in liver from the skeletal muscle lactate and the reuse of glucose thus synthesized in muscle for energy purposes. That is why it is called as Cori cycle. It takes place in the muscle. Next comes the glycogenesis. Name is there, glycogenesis, the synthesis of glycogen from glucose. It takes place in the cytosol. It requires UTP and ATP. So this is the cycle for your glycogenesis. When glycogenolysis, name is there, glycogen gets lysed to form glucose. So glycogenolysis. It is an irreversible pathway, very, very important, and it takes place in the cytosol. So glycogen forms glucose. Next is a very, very important pathway. It's called as hexose monophosphate shunt, or it is called as the pentose phosphate pathway, triple P. This is the alternative pathway to glycolysis and TCA for the oxidation of glucose. Here you use the biosynthesis of NADPH and pentoses. This is the reaction which takes place in HMP shunt. Significant pentose derivatives are useful for the synthesis of nucleic acid and nucleotides. Here NADPH is required, nothing but nicotinamide, adenine, dinucleotide, phosphate, hydrazide. Next comes the uronic acid pathway, or it is called as the glucuronic acid pathway. It is another alternative oxidative pathway for glucose, where you have glucuronic acid and pentose, which is being synthesized, nothing but ascorbic acid. Here you use UDP, uridine diphosphate. These are the references that I use for my seminar. Thank you.